So the next, in terms of alignment, there's something else we have to understand is how dissimilar are certain sequences. Okay, and therefore you're going to learn two, and there are many different distances, and they all come from math and are used in computational analysis quite a bit more in the IT sector. But there is some application for these what are called distances. Okay, so a distance, not as we conventionally understand it, but a distance can be used to measure the distance between the two sequences. And when we talk about the distance here, we are talking how dissimilar those sequences are, which is a measure of, the quality of your alignment, right? Or the starting quality of what you would need to make those sequences the same, or the minimum number of single edits to change from one sequence to another. Okay, and as you can see, this comes from the IT sector. Okay, and here's some examples here. So here's two sequences. They kind of look the same, but let's look at what a Hamming distance is. So I line the sequences up against each other. ATG is the same, the last T is the same, and everything else seems dissimilar. So I'm going to find all the dissimilarities, and remember the number of edits that need to be made, which means the number of times this sequence has to be changed in order to look like this sequence, or vice versa. And you'll realize that, you look at the mismatches, and there's 12 mismatches, which means 12 modifications would have to be made in order for these sequences to look to each other. So this is 12. Now this is a negative score, right? It's the distance. So you can tell that these are really dissimilar, because out of 16 elements in this sequence, 12 of them are dissimilar. Well, what about the Levenshtein distance? What about the Levenshtein distance? So in the Levenshtein distance, you'll realize that same sequence, same two sequences, just like this, S1 and S2, but what you're allowed to do is you're allowed to put gaps. And the moment you're allowed to put gaps, you see just a gap between these two lines up really well with parts of the sequences. And you're going to put gaps where it not belong. So now, because you're allowed to put gaps in a sequence which is actually quite similar but didn't look so when you just lined one against each other, you'll realize there's huge sections. So now you say, I'm going to find the dissimilarities. Same thing, the distance, the number of edits that have to be made. For example, C would have to, a gap, you'd have to put a C here, you'd have to put an A here. So the number of dissimilarities, or the distance, or the number of edits you would have to make this sequence look like this sequence, is 4. So the Levenshtein distance, which allows the flexibility of introducing gaps, tells you that these are a lot more similar than you think if you just line them up and said, well, this is not the same, right? And you can see that gaps in this case are called for, because there's huge regions which are very similar, and you don't get a sense of that because you don't add gaps, and therefore they appear this dissimilar. Okay, so here is an example of a Hamming distance and a Levenshtein distances, and again, it has something to do with alignment of sequences and their identities and their similarities.